Hey everyone, I'm Aryan Singh. I'm a rising senior at Troy High School. And I just want to thank, first of all, Hussein for being my mentor and leading me throughout this project. And my topic is MNR1 and its role in ovarian cancer. Yep. So let's just see the global prevalence of ovarian cancer. I'm sure you've seen a ton of ovarian cancer presentations by now, but this will be really quick. And as you can see, it's really prevalent in a lot of these more developed countries, and specifically in the US, nearly 20,000 women are diagnosed with ovarian cancer and 13,000 die. And a lot of the times when these patients are diagnosed, they're actually diagnosed at a very late stage, stages 3 and 4, where as you can see, the 5-year survival rate significantly decreases. This combined with its global prevalence makes it one of the most lethal of all gynecological malignancies, with a 5-year survival rate of 15%. So what is the standard care for ovarian cancer that we have? So most notably, it's surgery or removal of this cancerous tissue followed by a combination of chemotherapy. And while on the outside, it looks like 80% of these patients respond to this treatment strategy, really what we're seeing is within the first 5 years of the treatment, 90% of patients develop recurrence. So why does this overwhelming response to recurrence occur? Well, there is two main reasons. One of them is the micrometastasis of the movement of these cancerous cells across the body. And the second one is chemoresistance. So what is chemoresistance? Well, as you can see in a bulk tumor, there's two different types of these cancerous cells. They're the most sensitive and the more resistant cells. And these sensitive cells are going to cause a tumor shrinkage when they die to the chemotherapy, while these more resistant cells are going to reproliferate and metastasize, causing a tumor relapse. And the main characteristics of these resistant cancer cells is that firstly, they're anti-apoptotic, they avoid cell death. And secondly, they're metabolically adaptable. They can almost grow in any part of your body. So we want to look at cancer cell death. And as you can see, in a normal cell, there's a fine balance between pro-apoptotic and anti-apoptotic signaling. But what we find is in a cancer cell, it's heavily weighed to the anti-apoptotic side. And what I highlighted here is BCL2 and BCL genes, because these are two genes that we specifically work on in this lab. And what's to note about these genes is that they primarily act on the mitochondria, where they cause cell death through something called MOC, where they essentially poke holes through the mitochondria, lysing the contents to the cell and killing the cell. So one notable mitochondrial protein that we found was MNR1. And this protein was found to be characterized in lung and liver cancer. So we wanted to see its role in ovarian cancer. So before I jump to the gun on that, what is MNR1? So MNR1 is mitochondrial nuclear retrograde regulator 1, a lot of words, but we're just going to refer to it as MNR1. And it's a protein that has three main functions. It can bind to BCL cell in the mitochondria, where it serves as an anti-apoptotic phenotype. And it can also be phosphorylated by the ABL2 kinase and bind to COX4, which enhances cellular respiration. And thirdly, under low oxygen concentrations or hypoxia, it can act as a transcription factor in the nucleus, and it can regulate itself and even other genes. So having seen that it binds to COX4, our working hypothesis was that chemotherapy, specifically cisplatin, will cause this DNA damage, which will activate DNA repair kinases. And somehow these DNA repair kinases will phosphorylate and stabilize MNR1, binding to COX4. And we know that once MNR1 is bound to COX4, it increases oxidative phosphorylation, bettering our response to repair, essentially causing chemoresistance in these cancer cells. So before we jump into MNR1, let's just see, is MNR1 there? And the answer to that question is yes. It is differentially expressed across a panel of ovarian cancer cell lines. Specifically, we have low concentrations, we have high concentration, and we have some that are not detectable by Western. And the three cell lines that I've highlighted in these red boxes are three cell lines that we're going to be working with in future papers. So we have three main questions that we want to ask ourselves, or question 3 is more of 2 parts. But MNR1, is it regulated at the transcriptional level or post-translationally? So does it occur while it's being transcribed, or does it occur after it's already been a protein? And secondly, what does MNR1 bind to? We know it has 3 different sites, but in the cancer cell line, where is it primarily active? And the third question is, what is the functional role of MNR1? If we knock it out, what happens to the cell? If we re-overexpress it, what also happens to the cell? And essentially through these three questions, what we want to answer is an overall study goal is, can MNR1 be therapeutically targeted for the treatment of ovarian cancer? So question one, to answer this question, we set up an experiment between a low concentration of MNR1 cancer cell line and a high concentration of MNR1. And we treated with cyclohexamide. And essentially what cyclohexamide is going to do is going to pause all translation of all RNA. So essentially we're going to be left with the basal level of the protein, and we're going to collect it at 0 hour, 1 hour, and 2 hours. And what we saw was in the low concentration cell line R182, MNR1 within only 1 hour, almost 80 to 90% of the MNR1 has been degraded. While in the high concentration of MNR1, we see a stabilization of MNR1. So this answers two things. First of all, it is being regulated post-translationally, and secondly, it is differentially regulated across two different cell lines. So secondly, what does MNR1 actually bind to? So to answer this question, we did an IP, and while normally for an immunoprecipitation, we will pull down MNR1 itself. However, there was no antibody for MNR1 that can specifically work for IP yet. So we pulled down two known binding sites, BCLXL and COX4, and then checked later for their MNR1 expression. So what we saw was when we used the TKO cells and we pulled down BCLXL, we saw that MNR1, yes, is actually bound to BCLXL. However, when we pull down COX4, contrary to our hypothesis, we don't see that it's bound here, but we'll come back to that later. So having seen all that, our hypothesis is that MNR1 promotes ovarian cancer chemoresistance and reoccurrence. So firstly, what we did is we knocked out MNR1, as you can see successfully done here, and through some help, with some help from Nick, I was able to create a volcano plot of the knockout effect of the genes in the knockout cell. And what we saw was two specific genes that are downregulated that I highlighted here is THBS4 and COL1A1. And specifically, THBS4 is a family of glycoproteins that mediate cell-to-cell interactions, and collagen is a part of the ECM. And essentially, why we highlighted these two genes is because they have a strong role in the cell-to-cell interactions and how the cells actually aggregate and create tumors. And what Hussein has shown previously is that when you knock out MNR1, they're actually not able to form these tumors. So we're going to answer, what is the effect of the loss of MNR1? So we knocked it out. We saw it causes a different cascades of genes, and we want to see what is the effect on chemoresistance. So to answer this question is, we had two cells. We had the wild type, and we had the knockout of MNR1, and we treated with increasing doses of cisplatin, a very common chemotherapy, and we checked to see its survival rate. So firstly here, what we have plotted on the y-axis is our object sum area of thick spread. So this is just the confluency of the cell measured by the machine, and this is our time course. As you can see, already in the no treatment, it's growing fine. It can handle up doses of 0.6 and 1.25. It's growing fine, but all increasing doses, 2.5 and above, kill the cell. So what we calculated is the IC50, and just for context, the IC50 is the amount of cisplatin we need to add to kill 50% of the cells. So secondly, what we did is when we knocked it out, already in the no treatment alone without any cis, there's already a decrease in the growth compared to the wild type. And all subsequent increasing doses of cis kill off the cell. This makes it way more chemosensitive. And you can see from the IC50, it's only 0.003. That's how much cis we would need to actually kill 50% of the cells. So now that we saw that we knocked it out, we're saying, hey, if we can knock this out and we can reintroduce an MNR1 plasmid, can we rescue chemoresistance? And to specifically answer which binding site it works from, we did 3 different plasmids. We have BCLXL MNR1 plasmid, we have the COX4 plasmid, and we have a transcriptionally active MNR1, the MNR1 that acts as a transcription factor. And we did the same exact experiment with cisplatin and increasing doses, and we wanted to see, can we actually rescue chemoresistance? So firstly, what we did is we did a Western just to make sure post 72 hours of transfection, is MNR1 actually been properly transfected into the cells? And the answer is yes, we can see screaming levels of MNR1. And one other marker I wanted to mention is PGC1 alpha, because this is a gene that is, a protein that is specifically regulated by MNR1. So we should see an increase in the transcriptionally active one, which we do see here. So firstly, when we treated it with BCL-XL, we were able to see an IC50 of 6 micromolar, which if you can remember from the wild type was 1.8. So we were able to make these cells even more resistant than the wild type. And you can see they're surviving increasing levels of doses of cis all the way up to about 2.5. Following this, we also did one with the transcriptionally active MNR1. And again, we can see the same exact results where they're actually surviving higher levels of cis than they were before. And we also had one with the COX-4. However, because the cells were too proliferated and they were too confluent by the time we ran the experiment, we weren't able to get conclusive results for that, but we'll come back to that. So just to summarize all of the things I've thrown at you, MNR1 is firstly, it's differentially expressed across different ovarian cancer cells. It is regulated post-translationally, and the loss of MNR1 enhances the response to cisplatin or causes chemosensitivity. Therefore, we believe when MNR1 is bound to BCL-XL, it offers a anti-apoptotic function and therefore confers chemo resistance in ovarian cancer. So some things I wanted to mention about the COX-4 because we weren't able to let it work. So we wanted to retry to see if we can transfect it in a more stable cell line and see its effect in a seahorse machine where we could see its cellular respiration graphs. So one thing I wanted to mention is a lot of this was the wet lab, and I wanted to mention the stuff that I was doing with Nick. So welcome to, some people would say the boring side of biology or the dry lab. But I found this really interesting. This is kind of like the workflow I have in R, and you can see some pathway analysis I was doing. But to make it more intuitive for you guys, here's some of the bioinformatics stuff I was able to do with Nick. This on its own could be its own project, but I don't know how to explain all this just yet. But I was able to create a few things with differential gene expression analysis, the volcano, PCA, and heat map. I was also able to create a immune deconvolution and a few pathway analysis, SPIA and gene ontology and the KEGG pathway visualizations. So I just want to thank everyone who's helped me today. Specifically, I want to thank Hussein, who's led me through this project. I want to thank Nick, who taught me all the bioinformatics I know. I went from zero computing knowledge to that. And I also want to thank Dr. Alvaro for leading us on this project as well. And thank you all for having me here.